I think so. The next one we're going to cover is templates, which is one part about the data storage or the data acquisition side. And is that so maybe I can just first talk about what templates are. It's probably a little bit obvious, is that they're documents that have a predetermined layout and a specific style that allows reproducibility of new documents on the same exact layout, pattern, and guidelines. So this can apply for many different types of experimental data or reagents even. So if you have specific tubes that you're using with a series of experiments, you may want to write down a template for those tube labeling, like for example, the date, the genotype, or the specific conditions. And in terms of maybe microscopy data, you want to perhaps have a name template, like we mentioned earlier, with perhaps date and specific ontology words to include, to indicate the experimental conditions or the fluorescences, things like that. Yeah, so the obvious benefits of using templates is that they save time, they enforce the best practice, and they provide a guideline for users. And it gives you more confidence about the data. So sometimes maybe when you're so tired and you have a short time slot, you might make a mistake in your file name and save the wrong experimental file to the wrong name. And this certainly has happened to me. Yeah, so giving yourself that confidence is important for when it comes to using templates. And this also allows for automatization of searching and processing and analysis. So if you have standardized keywords, you can use it to search your files to find the ones that you are interested in. So in your templates, you can apply them in your electronic lab notebook. You can also, yeah, so like mentioned, you can do this for sample registration. So you write down for which samples and you have a specific template for the samples, for project structure, and also for your README file. So once you set up the template once, you can reuse it over and over again for different experiments or experimental replicates. Yeah, so you can do this for experiment type and techniques, and this is useful for anything that you have to repeat. So in the interest of time, I think I would just share the one that Jean-Karim had shown us earlier. So this is the checklist that Jean-Karim had shared with us. So if you see section 4, document data here, you can find template files that Jean-Karim here provided for images and arrays. So I would just click on this and you will see these are some of the templates for recording the metadata of images. So Jean-Karim, I don't know if you want to add something to this about yes. The YAML format.
Yeah, so there are two things here. So first, this one is mainly aimed at fluorescence microscopy type of images, because that's the things I've been mostly involved in. But you could adapt it in the more general way. But I want to point out mostly the bottom part, which deals with the experimental metadata I mentioned before, which are kind of important to capture, and the section about channels, which capture what basically the labels and the entity that are in each channel. And the choice of YAML format is meant to be a format that's meant to be both human and machine readable. So in principle, you should be able to use this and basically fill it out with a simple text editor. And it should be easy for a script to extract the relevant information in it. And I use this for basically converting the metadata to submit, for example, to the image data resource. So if I have this type of metadata coming from images, then it's easy to convert to what is required for data deposition, because this is the minimum that they need, but it's basically need to be associated with with images and slightly different formats. So that makes it easier to script than actually filling out manually some forms and spreadsheets.
Yeah, so thank you for that. I think as a part of a homework, maybe you want to note this down, is that you can look through your work process, your experimental data acquisition process, and your data lifecycle, and as well as the analysis side, and identify areas where you can use templates and start developing these. Yeah, so we'll just leave that as homework for you.
I just want to say, for example, one way of using that kind of file is to put it, for example, in the same folder as the images to which it applies. And the point also I forgot to mention is at the bottom, the associated data files, where you can describe, for example, the tabular data that comes out of the analysis of images. And then there's this other templates to actually describe the tabular data and give some information about it, that you can keep them together. So this is basically a low-tech way of doing data management if you don't want to use a database to keep those information.